This tutorial will show you how to solve the genetics problems regarding ABO blood types and the Rh factor. There are many different types of markers on the surface of our cells, but one of the most important ones is the one called the ABO blood types. So if you happen to have blood type A, what it means is your cells produce a antigen called type A antigen, and that is different from someone who has blood type B, who produces B antigens on the surface of their cells. If you happen to have both the A and the B antigens, then you would be considered to have blood type AB. And if you don't have any antigens on the outside, then you're considered to be type O. So these four blood types are controlled by one gene that we abbreviate I. And this gene involves two new concepts. They're different from the other autosomal genes that we've already talked about because this gene has multiple alleles, more than two, and it also shows codominant alleles. So there's actually three alleles of gene I, and they're called big IA, big IB, and little I. So remember, dominant alleles are the ones with the capital letters. So big IA and big IB are both dominant alleles, and the little I is a recessive allele. Someone with a genotype of little I would not make any antigens, and therefore their cells would not show either A or B antigens, and so we consider that to be a type O phenotype. Someone who is IAIA would only make the A antigens, and therefore they would express the A antigens on their surface, and they would be called the type A. But the heterozygous condition IA little I, in this case, the big IA allele is dominant over the recessive little I, and so they would still be the type A phenotype. So in a similar fashion, the IB genotype and the IB little I genotype will cause the cell to express only the B antigen, and therefore the cells will be considered to be type B blood. If someone has IA-IB genotype, that means that both the IA and the IB alleles are going to be dominant together, so they're called the codominant alleles. And both antigens will be made by the cell, and so the cell will be considered to be the type AB blood. This table shows the summary of all of the different genotypes that are possible to make the type ABO blood types. You will need to know this information in order to figure out the genetics problems. So we're starting problem 1. If Bob has type A and Alice has type B, what are the possible blood types in their children? So for all of these genetics problems, you want to figure out the genotypes of each parent first. And since Bob is type A, which is a dominant phenotype, he could be heterozygous or homozygous. And you want to look at the question to see if there's any information that would give you a hint as to which one he would be. Since there is no other information, we will assume that he's heterozygous so that you can see all the possibilities in the offspring. And same with Alice. So if Bob is heterozygous for type A, that would mean that he has IA and little I. If Alice is type B and heterozygous, then she would have IB allele and little I allele. So I'm going to have you fill out the Punnett square. So did you get this Punnett square? Now fill out all of the genotypes of the children and then figure out what blood types that would be. Did you get this answer? So when you look at this Punnett square, it shows that all 4 blood types are possible in their children. So you can have type AB, A, or B, or type O. We're moving to problem number 2. So if Joe has type AB and Jane has type O, what are the possible blood types in their children? So again, break it down. You're going to figure out the genotype of Joe and then the Jane's genotype and fill out the Punnett square. Pause until you figure this out. Hopefully you got this in your Punnett square, and now fill out the rest. So here we have 2 possible blood types in the children, either type A or type B. For problem number 3, I've set up a family of 5, and the husband thinks that the wife had an affair. So you're going to work out the ABO blood type for each child and try to figure out if there is one that does not belong. So you will need to figure out the genotype of each parent. Since Carmen has type A, again, we're going to consider her to be heterozygous. And for Carl, since he is type AB, he can only be he can only be IA and IB. The next thing you want to do is to figure out the genotypes of the children. So first we're going to fill in what we know based on what their phenotypes are. So son Clyde has type A blood, and therefore you know he must have one IA allele. And the daughter Colleen has type B blood, so you know that she has one IB allele. But for Calvin, he has type O blood, so he must be homozygous for little I. When we look at Clyde, he has one IA allele. If his father gave him the IA allele, then Clyde could have received from the mother either an IA allele or a little I allele. So we don't know if Clyde is homozygous or heterozygous, and that's the correct answer to put down. For Colleen, she must have gotten the IB allele from her dad, and therefore she must have gotten a little I allele from the mom. If she had gotten the big IA allele from the mom, then Colleen would have ended up being type AB, and that's not what she is. So the answer is Colleen is heterozygous, IB, little I. For Calvin, we already said that's a straight little I, homozygous. So what does that mean? Calvin had to have gotten the little I allele from the mother, but he was supposed to then have gotten the little I allele from his dad. But in this case, that is not possible. So Carmen must have had an affair, and the son Calvin was the result of that affair. Okay, hope this was helpful, and we're moving to something else. So now we're going to talk about the Rh factor, which also happens to be another antigen on red blood cells, and very, very important, especially when we're talking about blood donations and tissue grafts. So it's called the Rh factor because it was originally found in the rhesus monkeys. And this is the factor that is sometimes called the positive negative on your blood. So if you're A positive, that means you have the Rh factor and you have type A blood. And if you're O minus, that means you don't have A antigen, a B antigen, or the Rh factor. Okay, we're going to try a problem. So the gene R for your class is going to be the one that controls the Rh factor. And it's a straight, complete, dominant autosomal gene. So that means there's one allele, the big R, that is completely dominant over the little r recessive allele. So if you have someone who is genotype big R, or big R, little r, they will have a phenotype that's called the Rh positive, and their cell will make the Rh factor, which was an antigen on the surface of the red blood cells. But if they have a genotype of little r, then the cell does not make the Rh factor, and that person is considered to be Rh minus. So we'll do a problem now. If you had a Rh minus woman marrying a Rh positive man, what could their children have in terms of this Rh factor? So you want to go through and figure out the genotypes of the parents and then the possible gametes and fill out the Punnett square. So did you get this answer? I've only filled out one column because the second column would be exactly the same, but the children could be either Rh positive or they could be Rh minus. And in class, we would have already talked about that if you are a Rh minus woman, having an Rh minus child is no problem, but having an Rh positive baby can be a problem, especially for the second and third children. Okay. Hope this was helpful and that you're able to now solve the blood type problems. So make sure you try out the quiz and check to see that your answers are correct. And if you're still having a lot of problems, let me know.